Hello, guys. Everybody who's watching this, welcome to the fifth Journal Club. We have been doing this pretty regularly. I think the bi-weekly thing is good.
hmm
Regular for everybody. We have a periodic recurrence. I like this better. Um, Today, Maria will be presenting the article Suppression of Age-Related Salivary Gland Autoimmunity by Glycosylation-Dependent galectin-1 driven immune inhibitory circuits. What do you guys think about this article on the first glance?
First glance, honestly, I was like, oh my gosh, more processes, metabolic functions, all that. So I'm like, just give me the cellular work. But when you get into it, it's like you just appreciate so much more how much the immune cells are responding, in a like biochemical manner. So I thought it was very useful.
Yeah, I appreciated that we're talking about glycans, right? Because glycosylation, I think, is overlooked. We like to think of everything as important if they're proteins, and glycosylation and glycomics is just relatively recently starting to make some good headway in its role in especially autoimmunity, as we find in this paper, but disease in general.
Yeah, I recently found something cool that there is this protein called phytohemagglutinin. Have you heard of this? And this thing was initially used as an immune stimulant because it binds to galactins and not just galactins, a lot of glycosylated sugars. And lately, PHA is also used almost like an antibody to detect glycosylation. So you would use PHA and conjugate it with some fluorophore, and this itself is like an antibody that gives you an idea about cell glycosylation. Pretty cool stuff.
Yeah, generally they do something like this with different types of lectins.
Very cool.
I have very little knowledge about lectins, so I am looking forward to this presentation to educate myself a lot.
Yeah, same. Definitely outside of my comfort zone as a viral immunologist to talk about glycans and autoimmunity and diseases I can't pronounce. So it was a great paper to read over.
Yeah, I had to remember this name, Sjögren's disease. Sjögren's, the not the weird name that I always thought it was. Anyway,
You're so good with names.
yeah, except your name, and now I'm getting the hang of that.
Gosh.
All right, Maria, let's begin.
Okay, so hi everybody. My name is Maria, and today I'll be presenting a presentation about galectin-1 and its role in Sjögren-like disease in mice and also in Sjögren's syndrome in humans. So first, let's talk about glycosylation. As we probably know from our previous classes, glycosylation is a type of post-translational protein modification. This process takes place in endoplasmic reticulum and in Golgi apparatus. There are many enzymes involved in it. It influences protein folding, structure, and function. It may impact surface expression and also secretion by the cell function. It may, it can impact catalytic functions because of the folding. Okay, and the whole process is very important, which is like pictured by the fact that about 1 to 2% of the genome is responsible for all the proteins involved in this process, which I think is quite a lot. Okay, and what is also important is that the whole process is different in different cells, tissues, organs. It can change during the life of the organism and during, for example, infections, pregnancy, different diseases, and it can also be influenced by vaccinations, for example. Okay, so when it comes to glycosylation and immunology, so it's the glycosylation of proteins definitely influences receptor expression and clustering of these receptors on the surface of the cell. It may alter antigen presentation, as different glycosylation of major histocompatibility complex proteins may influence their binding to the antigen. Also, it impacts cell migration, because some of the receptors, when they have altered glycosylation, they bind differently to their ligands, so the migration is also influenced. Also, it's worth mentioning that ABO blood types are basically glycans on red blood cells. The patterns of glycosylation on different cells take part in self and non-self identification. And these patterns are generally called glycocalyx. And what was found was that changes in antibody glycosylation, especially in FC part of the antibodies, may alter the nature of the reaction triggered by those antibodies and their therapeutic efficacy. So generally, if you want to research into antibody glycosylation, there are many articles that have researched this part. So to sum up this part of presentation, glycosylation has a role in immune homeostasis and also acts as a rapid regulator of immune response, because glycosylation is very like prone to changes in environment. It is the fastest probably regulator of protein expression.
I want to add something here. For me, the hardest part about understanding glycosylation is that I cannot imagine, like, physically what kind of thing it could be doing. Like, if a glycan chain is attached to a protein, it becomes very difficult for me to visualize how could that be changing. Maybe what you said, changing the conformation of the protein, just adding something very heavy, maybe that's how these things do, or maybe there could be something else.
Because generally, glycosylation is a process of adding glycans to amino acids. So proteins interacting with each other is basically interactions between amino acids. So I guess changes in, moieties added is generally changing this this binding, I guess.
Yeah.
I'm a biochemist, so I'm also not sure what exactly happens there. Okay, so one of the proteins that take part in those processes of recognizing sugar moieties, let's say, is galectin-1, which is a type of lectin. It is expressed by immune, endothelial, and cancer cells. When it comes to cancer cells, it plays a role in cancer cell escape from immune cells. This protein has characteristic ligands, which are, let's call them LACNACs. These are N-acetylactosamines. And they are mostly expressed on membrane glycoproteins, for example, CD4, CD7, and others. The funny thing is, unlike many lectins, it is secreted, but it doesn't have this secretion signal. So it's secreted via NAK pump. So it's quite unusual, I think.
Hold on, what does that mean? I mean, how are the pumps secreting these?
So it's like during the signal in nerve cells that is just like electric potential. So I guess it's somehow caught in this exchange of ions in the cell. So I think it's very funny because all the proteins are secreted specifically because of the secretion signal on them, and this one is not.
Yeah, that's pretty strange. You could there are also other things. Have you guys heard of these proteins called alarmins?
Mhm.
So alarmins, these are cytokines like interleukin alpha 1 alpha and IL-33. These are just like cytokines, but they don't have a secretion signal as well. But the way they get out of the cell is through cell damage. So if they're outside the cell, they're like danger signals. But for this one,
Eh,
it
and that makes like perfect sense, because when you think
mhm,
about something like psoriasis, and you have rapid, proliferation of keratinocytes, but then they die as well, and you have a lot of shedding and plaque buildup and so forth. These cells then express IO1, which is a huge marker for this type of pathology. So
mhm.
I can see that.
Yeah, so for alarmins, if it's cell damage, I think I cannot get myself to imagine how the pumps are, because the pumps have a very narrow space for the ion transfer, so there is no way the galactins are going through the pump.
I found it in one of the articles, and I also found it was quite funny. And
Yeah.
they just said it didn't have a secretion signal, so well, I thought maybe this pump is a little bit bigger for the protein.
Mm-hmm. You mean even smaller for the protein?
Yeah, smaller, yeah. Okay, so coming back, what are the effects of galectin binding to its ligands? So there are two groups of those effects. We can call them T cell related, and these are apoptosis, which is selective for Th1 T cells and Th17, and also cell turnover, immune regulation, and cancer evasion. And also non-T cell related, which are adhesion, B cell development, mRNA splicing, angiogenesis, and many, many more. There were also like nerve and muscle development, but it's not related to immunity, so not relevant now.
Wait, I guess that's still very much related. The adhesion and mRNA splicing, maybe some way indirectly, that is all related to immunity too. Adhesion, especially.
Yeah, that's true. It acts as a negative regulator of immune responses, as it generally suppresses immune response. It also acts during development of immune cells. Its expression in thymic stroma plays a role in deletion of autoreactive lymphocytes. And also it's expressed and secreted by activated Tregs, which also confirms its role as a negative regulator. Okay, so here we have many of the ways galectin may act on immune cells. So generally, what are the effects is that it promotes resolution of autoimmune inflammation, may suppress allergic inflammation, promotes fetal-maternal tolerance, so it also is important in pregnancy, favors tumor immune escape, and compromises antimicrobial immune responses. So, well, generally, it acts as anti-inflammatory agent. Okay, so now quickly I will tell you something about Sjögren syndrome. It's an autoimmune disease which is characterized by infiltration of leukocytes into of salivary and lacrimal glands. And the effects for patients are dryness of mouth and eyes, which I can only imagine is very unpleasant to live with. When it comes to the causes of this syndrome, well, scientists found that it may be genetic predisposition, probably like with other autoimmune diseases. And they found that HLA-DR and HLA-DQ haplotypes are more prone to developing this syndrome. There are also environmental factors. Among them, viral infections were mentioned. And why is that? It's probably due to molecular mimicry of some viruses. And these viruses then cause production of autoantibodies. They mentioned HCV, EBV, and HTLV1. But probably not in every person it causes this syndrome, and also there may be some other environmental factors that influence.
Yeah, when we're talking about viral infections, I want to add something here, that a lot of autoimmune responses and diseases like rheumatoid arthritis, rheumatic diseases like rheumatoid arthritis or lupus, there is an autoimmune response that mimics a lot like an antiviral response. These B cells and T cells, they're responding to TLR7, which responds to single-stranded RNA, usually found in the case of viruses. They're making a lot of anti-nucleic acid antibodies. They have a lot of cytotoxic T cells whose major job should be to be killing viruses, but then now they're going and killing your own cells. So viral infections and autoimmune diseases, they're very similar in some cases. It's just that autoimmunity is like a viral response, but without the presence of a virus. And probably it's more in certain autoimmune diseases than other, like you said, Sjogren's. Not everybody has that.
Probably. I had a friend who, well, is quite young for RA because she was like around 20 when she developed it. And she said that she probably had genetic predisposition because her aunt also has RA. But probably in her case, it was triggered by very strong bacterial infection. Okay, another factor that may cause or may play a role in Sjögren syndrome development are hormones. What was mentioned was estrogen, but for me it was a little bit elusive, as some researchers mentioned that like estrogen may cause Sjögren syndrome, but others said that using agonists of estrogen receptor helped to cure, or maybe not cure completely, but alleviate this immune response in Sjögren syndrome. So
is this disease also like more prevalent in females?
Yeah,
you know?
Females are 9 times more or 10 times more prone to this disease.
Oh man. We just discussed in our latest podcast about this X chromosomes doing something weird. What? And yeah, probably something is really the key in that X chromosome, that extra that females carry.
Yeah, I also think that hormones are quite important regulators of immune response. So maybe also hormonal factor is there.
Yeah.
Okay, so what exactly happens with immune system and those cells in glands in this disease? So first, let's concentrate on epithelial cells in glands. Those cells release chemokines and adhesion molecules that promote migration of dendritic cells, T and B cells. And what is quite interesting is that they could act as antigen presenting cells because they start expressing some molecules that are typical of some of the APCs. So we have CD45, CD40, MHC1, and TLR2 and 3 and 4. Okay. What is characteristic is that DCs, mostly PDCs, infiltrate the glands. Then they produce interferons, which causes then more lymphocyte retention and activation, and also BAFF production, which is very important for survival of mature B cells. And then it causes further autoantibodies production. So normally those self-reactive B cells would be deleted, but due to the BAFF overproduction, those abnormal B cells maturate and produce autoantibodies.
Here, like when you say epithelial cells and glands pretty acting as APCs, I just want to appreciate how flexible everything is in our body, that things can take on each other's role whenever wanted. Somebody I was talking to, I don't remember who exactly, but I think it was in a seminar that somebody said you put interferon gamma to any cell type and they will start presenting MHC2 on that. So I don't know how true that is. I haven't checked, but you just feel like a lot of cells can in fact act as APCs if they're just given the right conditions. And epithelial cells, they're everywhere. So if they can present antigens, then you have this whole new places where T cells can be activated.
Yeah, that's true. Okay, so it's also characteristic that T cells infiltrate glands, and these are mostly CD4 plus T cells, but not only. And those T cells destroy epithelial cells. They promote apoptosis and act via cytotoxic agents like perforins and granzymes. And because of this destruction, we can observe those clinical symptoms like dryness of mouth and eyes because saliva and tears aren't produced. We can also observe abnormal cytokine production. In the early stages of the disease, there are more Th1 type cytokines like interferon gamma and interleukin 2. In the later stages, there are more Th2 type cytokines like interleukin 6 and interleukin 10. And what was surprising for me was that TGF beta is present in this disease. And as we all probably know, TGF beta is more anti-inflammatory, sorry. So it was quite surprising for me, but apparently it may be like the last way of our organism to prevent inflammation. So this cytokine is present. And also what I found is that in some diseases, like in systemic sclerosis, TGF beta acts as pro-fibrinous agent. So it may later promote damage in the glands.
Yeah, that's another function for TGF beta, that I think it causes the connective tissue to expand, and that's why you see these fibrous tissues. Yeah, maybe that's why we don't use TGF beta as a therapy for a lot of diseases. I don't.
Not alone though. So you have to remember the environment that TGF beta, would be in, because that coupled with something like IL-10 can lead towards, maybe regulatory T cells or Th17 type of pathology. So, you have to keep in context the environment that it's in. You know, you don't want to just say, let me go ahead and inject somebody with TGF beta in
Yeah.
and see what happens, but it has to be coupled with the right environment.
Yeah,
Yeah.
right. And as Anthea said, IL-6, that's a very important one, because if you give IL-6 and TGF beta, you don't get Tregs, but you get T helper 17 cells out of the naive T cells. So that environment is indeed very important.
Okay, and what is also typical are the autoantibodies, and the most like characteristic, let's say, are anti-SSA row. So they target row 52, which is a negative regulator of IRF activity. So if those antibodies are present, then the pro-inflammatory cytokines are elevated shortly. And also you can find different autoantibodies in this disease. So they're like more typical of lupus, for example, or there are also, you can also find sometimes rheumatoid factor. It's also quite interesting. Okay, so now I finished my introduction. So let's talk about why those researchers decided to examine galectin-1 in Sjögren disease. So generally, homeostasis in immune system is very important. So we both need active immunity in viral infections, but we also need self-tolerance so as not to destroy our own body. In this whole homeostasis, there are many receptors, pathways, transcription factors, and cytokines. And in all those processes and in all those elements, glycosylation plays a big role, especially glycosylation of the receptors, and also receptors that can bind glycans.
Do you know CD25? That's one receptor that is heavily glycosylated, and if CD25 is not glycosylated, then it is not retained on the membrane. So it
Yeah.
just, I think it just goes back inside the cell. So
Yeah.
that lets me imagine these glycans and some sort of anchors, like they're holding on to the membrane and making sure the receptor stays there. Pretty
Yes,
cool.
and also those glycans may help to those receptors to cluster, and as we know, some receptors need to cluster to function properly.
Yeah.
Okay, and as those researchers wrote, previously they found that this galectin-1 may play a big role in autoimmune diseases. So there were some previous articles that suggested that mice lacking galectin-1 or its ligands are more prone to autoimmune diseases. And so they wanted to examine it. Okay, so what they did was they bought mice, and they used only female mice, probably because Sjögren's disease is occurs much more in women than in men. And so they used mice knockouts, LGALS1, so they didn't express galectin-1. Those mice were knockouts of enzyme that produces branched glycans that are ligands to GAL1 protein. They also used non-obese diabetic mice, which are known to develop diabetes when they are aged, and also other autoimmune diseases like encephalomyelitis or Sjögren-like disease. And as control, they used wild type C57BL6 mice and bulb C mice. To examine the amount of autoantibodies, they used ELISA, and they assessed these levels of autoantibodies in mice sera. They also assessed the GAL1 in mice sera. When they were culturing cells, they were also assessing some cytokines, and they also used ELISA to do this. To detect autoantibodies, they also used HEP2 cells and immunofluorescence, as these cells are known to secrete many types of autoantibodies.
I'll just add one thing. So I recently did a journal club where I had to read about HEP2 cells, because I had no idea why would they be using a weird cell line, and turns out this is a carcinoma cell line that expresses about 50 plus autoantigens. So you just throw your sera onto these cells and add some secondary antibody with the fluorophore, and they will start glowing if any of the antibodies bound to their antigens. So they're very good for an autoantibody detection standpoint.
Yeah, that's very cool. I think you don't, well, if you don't know which autoantibodies specifically you have, you can just check if there are any using these cells.
Right, just anything.
Yeah.
It's like an array, right? It's like almost an array of all antigens, but a real cell is presenting to you all those antigens. It's very cool technique.
Yeah. They also did a lot of histopathology experiments. So what they did with those like tissue biopsies was they fixed them, stained them using hematoxylin and urosine, so typically, and they assessed inflammation score using Chisholm and Mason. Sorry, I don't know if I'm pronouncing correctly. Which is a scale of 0 to 3. 0 means no infiltrate, so no inflammation, and 3 means like more than 50 mononuclear cells per 50 mononuclear cells per 4 square millimeters. So okay, that's well, the scale used to assess Sjögren syndrome. And they also counted salivary ducts, as the number is correlated with the syndrome. In many experiment parts, they isolated cells from submandibular glands, lymph nodes, and spleens of mice, and they assessed surface markers, FOXP3 expression, and intracellular cytokines levels. They did it using flow cytometry. They also performed RT-qPCR to assess levels of mRNA of LGALS1, CXCL10, and CXCL9, and they also used GAPDH as control. And probably did it mostly in sera, and I also think that in those epithelial cells. They also decided to isolate dendritic cells from aged LGALS1 knockouts and aged wild type mice. And they cultured them with CD4 plus or CD8 plus T cells isolated from young wild type mice. And they cultured them in the presence of activating factors, so anti-CD3E antibodies. And after performing this experiment, they decided to mix these cells and media from this culture of CD4 plus with disease and culture them with cells from CD8 plus mixed with disease. And they performed it in the presence of anti-CD3E and anti-CD28 antibodies. They assessed proliferation using Vibrant's CFDA-EC cell tracker kit using flow cytometry. And one of the last things they did was they assessed salivary gland biopsies from patients and controls. So there were 19 patients with confirmed primary Sjögren syndrome and control subjects, which were actually people that had suspected CL adenitis, but not confirmed after biopsies. So here we have those inflammatory scores. So they mostly had those grade 3 or 4, and control subject, of course, had 0 inflammatory score. They were mostly women, mostly over 40 or even 50 years of age. And those primary Sjögren syndrome patients were mostly diagnosed with anti-RO antibodies. And what they did with those biopsies was they stained them using anti-gal1, anti-CD8, anti-CD4, and anti-CD20 antibodies to check the expression in the salivary glands. And the last thing they did was they treated old NOD mice using recombinant galectin-1 or PBS. They did it every 2 days for 2 weeks. After that, they sacrificed the mice, isolated salivary glands, and also isolated the cells. They stained them and compared between those 2 groups.
It's weird that these mice models of autoimmunity, they don't have a single kind of autoimmunity, because NOD, you said they're using here for salivary gland inflammation, but NOD is also a model for type 1 diabetes. And same here, there's another model for lupus called MRL-LPR, but those mice also develop a Sjögren syndrome like thing. It's as if you have one break in tolerance and everything in your body just goes wrong. So these mice, they don't develop one thing, they just develop everything together.
Yeah, it shows that, well, there are some, well, let's say immune checkpoints that if they are wrong, then everything goes wrong, I guess.
Yeah.
Okay, so now let's go to the results section. Okay, so first let's look at those LGALS1 knockout mice and their characterization. As we can see, compared to a wild type mice, they had elevated anti-dsDNA, anti-nuclear antibodies, and also anti-SSA elevated. So everything went up. And using those HEP2 cells, we can also see that the autoantibodies were present because those cells, well, after using those fluorescent antibodies, they were glowing. And here we have this mean fluorescence. Okay. Okay, so, oh, what happened here? Okay, it wasn't supposed to look like that. Okay, whatever. Yeah, probably it happened when I downloaded the presentation. Okay, sorry for that.
It's all right.
Okay, so what else they did? They isolated salivary glands from LGALS1 mice and MG85 knockouts mice. And well, I wanted to compare the outcome. So we can generally see that there is immune cell infiltration in both L-GALS1 and MGAT5 compared to wild types. So this is generally this representative picture. And also the salivary duct count is also elevated. So we can see that those spaces are those ducts. So there are more of them in both knockouts than in wild types. And it's also here on the charts that this inflammatory score is well, generally much higher in both mice than in wild type.
Do we know how they calculated the inflammatory score? I meant to go back and look in the methods to see.
Yeah,
it was scored by a histopathologist.
Oh, okay. They were fancy.
Home and Mason score.
Yeah.
Is it 0 to 3? Because I remember seeing a 4 somewhere.
Oh, maybe to 4. Maybe it was my mistake. Yeah, it's to 4. Sorry for that.
Oh, okay. So they actually had a pathologist do it. Wow. And I wonder how they did it then. I wonder how, like, what would you count for? I'm sure you maybe have to zoom in and.
Yeah, so histopathologists have to do obviously a very specific sort of training. And when they do that, they learn exactly how to score these kinds of things. And they're obviously going to be blinded to it. So the researchers themselves aren't going to score in order for it to be published anywhere important. And then they score them blindly and then compare. So they're looking at cell infiltrates, inflammation, anything that shows up with whatever stain you're using. So for here, they're using H&E. And then they just score it based on their magic, the voodoo that histopathologists do. I don't know. I mean, it's so funny. Whenever I look at histopath, if there's nothing that's extremely obvious, I have no idea what I'm looking at. So it's always funny whenever they include them and I'm supposed to be like, yes, that purple tissue is a little bit more purple than the other purple tissue.
I trust him. I trust the pathologist.
Exactly. I blindly trust the pathologists.
Yeah. But what is also visible, I guess, for everyone without being a histopathologist is that the weight of the salivary glands was much bigger. So it also
I can measure that. Yeah. I feel confident.
I can scale like it's no one's business.
But I like this figure. It tells you that without galectin-1, you have higher inflammation score, but also without MGAT1, which is the enzyme that provides lactosamine, also have higher inflammation score. It doesn't tell you that they are both related or not, but both are probably required.
Yeah, it's probably that if there is no receptor or ligand of this receptor, then the whole signaling goes off. So yeah, I guess the whole pathway is quite important.
So one thing that I thought was interesting is this came out of PNAS, right? And they got these mice from commercially, but two of their figures are basically validation, partially, two parts of these figures is validation of what they purchased from the supplier, right? So when they're showing like figure 1A, where they show that they do in fact have significantly higher anti-DS, DNA, ANA, SSA, and then they show the histopath, they're just kind of validating that the mice that they're looking at are legitimate. I wonder why these had to take up two figures and they couldn't have just said kind of in a supplementary aside, like these mice perform as we expect. Now, they did look, and you'll get into it, whatever the rest of the immunophenotyping with the CD8s and the CD4s, but I thought it was interesting that some of them at least take up space with basically validating what they purchase commercially.
Or maybe probably they wanted to check if those mice have those Sjögren-like disease, let's say.
Yeah.
Like, is it a new thing that NOD mice develop Sjögren syndrome, like Sjögren-like?
I wouldn't expect it. I mean, maybe it is, and that's why it was included as an actual figure. But I don't know, just an interesting thing to take up space that you would, I mean, I just assumed that these Elgal's mice were well described if they were purchased commercially, but perhaps they weren't. And I'm just assuming that they had been done before.
Oh, you mean Elgal's knockouts?
I'll just,
we had 5 mice were actually purchased from Jackson, and I guess Elgal's one were from one of the researchers from France, I guess. So maybe they had to check those ones.
yeah.
But I don't know why those ones.
Yeah, a very random guess, but if I had to bet, I would say the Elgals in terms of SS is not known. That's what they write in the introduction as well, that we don't know, just in case of Sjögrens, how bad it is. So that's why they have these salivary gland scores specifically.
Okay, yeah, gotcha.
Okay, so as we see, their salivary glands look like they were expected to look, let's say. Okay, then
very crappy salivary glands. Very fine.
Okay, then they isolated cells from those salivary glands to check what cells were the components of the infiltrates. So here we have the gating strategy. It was like one of the more representative chart. And here we have the mean counts. So we can see that when it comes to these 2 mice, there are some differences because Elgas1 knockouts have more CD45 plus cells infiltrating the glands and more.
I'll just like to stop here for a second.
Yeah.
Do you think we should, briefly, just look at the gating strategy, what that means, like what kind of cells they're looking for? Anybody wants to lead that discussion?
Okay, so firstly, maybe let's look at this chart. So here we have, while using flow cytometer, you have at first 2 main things that you check. You check forward scatter and side scatter. So forward scatter is basically the cell size, and side scatter is the granularity of the cell. And then you can stain your cells using special antibodies that bind to the receptors that you may be interested in. So here they chose those CD45 and they decided to use it against this forward scatter. I think that they just knew that all those others are not lymphocytes because the size was probably off. So they just knew that around this place, there should be those lymphocytes, right?
Yeah. So apart from the size, CD45 is also a marker for myeloid and lymphoid cells. DCs have it, macrophages have it, T cells have it, B cells have it. I think natural killer cells also have it. So by gating on CD45 positive cells, they are just grouping all of these lymphoids and myeloids together.
Yeah.
And then they go for the CD3, which is specifically
lymphocytes.
Yeah, specifically T cells.
So
now they're specifically making sure that CD3 positive, CD8 positive, because it's something very strange. CD8 is also expressed by dendritic cells. So if they don't look at CD3, then some of the CD8 positives could also be dendritic cells. And they do the same thing for CD4, so that's our helper T cells. And that B220 is just CD45, which is a very heavily glycosylated form of CD45 that's expressed on B cells. So I think they're just generally grouping helper T cells, cytotoxic T cells, and the B cells with this floating gating scheme.
Yeah, I appreciate that they put a representative flow diagram on there, because whenever I get flow data that doesn't look gorgeous, I get embarrassed. But this, I mean, looking at the very first one where they have the CD45 forward scatter, and it just kind of looks like a bit of a mess, and they were still able to delineate that population. makes me feel a little bit better.
Yeah.
When doing you guys' initial gating for flow cytometry, has anyone ever tried forward scattered versus forward scattered? So forward scattered A versus forward
Sports their height to remove doublets. Yeah, I think that's pretty standard. I would be surprised if they managed to publish this and they didn't do
Yeah.
doublet exclusion.
Yeah.
Right. That's
Earlier
probably
on,
in a supplementary
First
figure.
thing we do at our lab, so yeah.
yeah.
Pretty common.
Earlier on, when I was beginner flow, and I don't think I'm an expert as nearly as much as people who do like 50,000 different strategies to get one.
Yeah, the 40 color panel. Yeah.
40 color panels, but I originally just learning flow in the beginning, we kind of just gated off of the dead cell population from just forward and side scatter, you know. So they're really progressing just how to get rid of some of the background noise, so your in gating will be a bit more cleaner. So
And usually if you're looking at a live cell population and you're performing it right, I mean, in general, doublet exclusion should only eliminate 2 to 3% of your live cells. You could probably get away without doing it, but still cleans up your data. But I don't know if it would, how substantially it would.
Depending on the tissue, though, right?
Oh, that's true. I guess I usually just work with cells, tissue culture.
Yeah.
For cells that don't stick together, or they should not be a big problem for doublet discrimination. But if you take out cells from the lamina propria and they are still stuck with the epithelial cells or whatever, then you have to make sure the doublets. And I would assume you would have a lot of doublets there if the straining, the cell filtering wasn't done properly, because those double cells will give you double signal of everything, and now you would think that you have high expression, but it's just two cells of the same things.
Okay. So as we can see, we have generally a lot of immune cells in the salivary glands of L-Gauss1 as compared to wild type. And among these, cytotoxic T cells are mostly elevated because others like T helper helper T cells and B cells are not elevated or not significantly. And when it comes to those other knockouts, all cells are elevated. So we have here a little bit of a difference between those two knockouts.
But that's probably because there are other galectins that also recognize sugars.
Yeah, probably.
Right? They go on to say that, I'm pretty sure.
Because I would expect MGAT5 knockout to have a more severe impact than just galectin-1.
That's true. Okay, and then they concentrated on those L-Gauss1 mice and in those infiltrating CD8 plus T cells. So, okay, again, the presentation here is bad. I don't know what happened here. But here we have the gating strategy. So they decided to check if those CD8 plus cells express interferon gamma, interleukin 2, or both. And so they gated for those CD8 positive cells, and then they checked the expression of interferon gamma and interleukin 2. I think it was this intracellular staining. And what we see is that there is elevated expression of interferon gamma. So there are more cells that produce interferon gamma and more cells that produce both interferon gamma and interleukin 2, but not interleukin 2 only. So it seems that interferon gamma producing cells are more important here. Okay, so another quite interesting thing they did was they checked PD-1 expression by CD8 plus cells. Should I talk shortly about PD-1 and PD-L1?
Oh yeah, for sure. Any markers you feel like are not super common, like they're not taught in an immunology 101 class, I think we should always try to talk about them.
Okay, so PD-1 is program death protein, and it's expressed generally by immune cells. And when this protein binds to its ligand, so PDL1, then those cells, it promotes apoptosis generally. So it's a way of stopping the inflammation. So when there are too many immune cells and the tissue decides that there is too many, it starts expressing PDL1, but not only tissue. And then the cells are apoptotic, and that's a way of acting anti-inflammatory. So generally like this. Do you want to add anything, guys?
So we may have heard PD-1 called an exhaustion marker for your CD8 positive T cells.
Oh, okay.
So yeah, just that whenever I think of PD-1, I think of exhaustion, T cell exhaustion.
I wonder if it's only an exhaustion marker when there's a lot of PDL1 around. Because PD-1 is also a marker for activation, because T cells only upregulated when they are activated. But I have heard that it's also an exhaustion marker. I'm not sure how can we draw that line between just say calling them activated or calling them exhausted?
Oh, I wonder if there's another marker that gets down-regulated when it goes from just activated to exhausted. I don't know. Be interested. I'll have to read more about that. I mean, I haven't really looked at PD-1 since intro immunology. It was a cool marker to look at.
Yeah. This
Maybe
is specifically
one of the podcasts.
Yeah, they specifically use PD-1 to mark T follicular helper cells because they express a lot of PD-1 when they are activated.
And it would make sense, right? If you have increased interferon, you would have increased PD-1. So the more inflammatory, the more likely the cells are to exhaust because they're constantly churning out pro-inflammatory cytokine.
Okay, so what we see here is that there is elevated proportion of CD8 plus cells that express PD-1. So it would suggest they are subject to exhaustion, and they are more potent to apoptosis. But what they also checked was that the expression of PDL1, so the ligand, in epithelial cells in the glands was downregulated. So, it actually was like those CD8 plus cells met like anti-inflammatory environment in those glands, in the first place. So even though those CD8 plus cells could die, they actually are not dying, let's say. Okay. Okay, so then they decided to check for chemokines. And so they first they checked mRNA expression in glands of CXCL10 and CXCL9. And it was elevated in Elgal's knockout when compared to wild types. So it suggested that epithelial cells in glands, they send this signal to immune cells that they should come there. And they also examined expression of chemokine receptor in CD8 plus cells in both L-gal's knockout and MG85 knockout. So in those CD8 plus cells, the cells that expressed this receptor there was a higher percent of those cells. But not in MG85, but we can say that the tendency is on the rise.
The naming convention for chemokine receptors is the worst. It's a nightmare, and not because of the structure like CXC or CC. It's that you would think CXCL3 is the ligand for CXCR3, but that's completely not the case.
Mm-hmm.
CXCR3 has the ligands CXCL9, 10, and 11. And for somebody who's new to this field, it would be so daunting that it just exhausts you after you learn a lot about these weird names. Because in cytokines, you have interleukin 2 receptor is interleukin 2 receptor. Pretty straightforward, not for chemokines.
So fortunately, the chemokine that I work on is fractalkine, CX3CL1, and it's the lone chemokine of the CX3CR1 receptor. So I don't have to worry about that problem for the most part. I actually had a question about this figure. Does anyone know why they would look at CXCL10 mRNA as opposed to the rest of the data, which is flow?
So CXCL9 and 10, I think they would have to do intracellular cytokine staining for that.
Yeah.
Right, which they also did for interferon and IL-2, right?
Yeah, probably something strange. So CXCL10, I want to tell you, CXCL10 is a very strange one. Just this one, because it's actually a cationic peptide. So it binds to DNA, and so this thing actually binds to DNA and all negatively charged things. So maybe that's why it acts very weird around antibodies. Could be
they
that.
have the CXCL10 conjugated antibodies.
They do? Where are they using those for?
Let's see. BioLegend sells them.
I was going to say BioLegend. They have everything.
Yeah, probably doesn't work as well. Some proteins, some antibodies, they just don't work as well inside, and especially intracellular staining. That's a tricky one.
Oh God, yeah. No, that's definitely true, and that may be. It was just an interesting thing that I noticed. Sometimes I don't know why they use mRNA expression in some things, and then in other things they're showing actual protein. And I know that there's whenever you're looking at a paper and you're seeing mRNA, that you can't necessarily say just because transcript's there, the protein's there.
Yeah. And all of these, the chemokines and the chemokine receptor, they are all downstream of TBET. And TBET is that transcription factor that gets activated by interferon gamma and IL-12. So this makes complete sense. You have high interferon gamma, you would also have higher these chemokine receptors and chemokines running around. Harrison, I just checked for the exhausted T cell thing. So it looks like the PD-1 is expressed in medium amounts on activated T cells, but very high on exhausted. And they also decrease some other markers that are regularly found. CXCR3 actually is a marker that they don't express if they're exhausted.
Oh, okay.
It's quite interesting.
Yeah, thanks for that. That's neat.
So next, they isolated DCs, dendritic cells, from submandibular glands. And first, they assessed the percentage of CD11C plus T cells and DC cells, and they found that the percentage was lower than in wild-type mice. And then they checked for the phenotype. So what they found was actually, even though there were less of these DC cells, they were actually more mature because they had elevated CD86, CD40, and MHC type 2.
Anybody wants a trick question?
No, but hit me.
Okay. So I was always taught that 80 and 86, they're almost synonymous, do the same thing.
Uh-huh.
But I recently read that one of them does a very, very different job, and that is 86. 86 is very highly excitable. It it binds to CD28 much better than 80, and CTLA4, which is an inhibitory marker that binds to both 80 and 86, that actually has lower affinity for 86. So if the APC is high on 86 and low on 80, it's less likely to be affected by CTLA4.
Yeah, I always thought 8086 was like
Mario
the way
Brothers.
it goes. Yeah, exactly. Yeah. Do you guys know why they did they present the data as MFI for the CD8086, CD40, and not for CD11C? Is it still flow that they're doing this on, I assume?
Yeah.
Yeah, it's because the percentage of cells expressing this, and it's like the this mean of the expression of those
So
receptors on the cells.
in B, are they first gating on DCs and then looking at their
Yeah,
86 MFI? Okay.
yeah, I think that that's what they looked particularly on, probably on those CD11C plus.
But why would they present the data as mean fluorescence and not as percent positive as they had done previously?
Probably because they wanted to show that, well, every cell probably is more mature. I don't know. It's not probably because of the percentage of mature cells, but that they, well, generally were more mature. I'm not sure, but that's what I thought while
So in
reading.
percent positive, it depends a lot where you're drawing the gate, and if you have a very loose kind of gate that includes everything, you may not even see differences.
Right.
But for the expression MFI, even if you have a large gate, you will know if the population is skewed towards higher or lower. So the MFI is very useful in that case.
It's just interesting to see both percentage and MF5 presented right next to each other, and they make kind
Yeah.
of claims on CD11C as well as CD86 and 80.
Yeah, I recently did an experiment. I looked at Tregs. We did not have any differences in Tregs, but we found very high FOXP3 MFI with a particular treatment.
Mm-hmm.
So yeah, we did report both of those just to show that yeah, we don't have more Tregs, but we have higher MFI in whatever Tregs we have.
I'm reading some literature where it talks about because the fluorescence intensity increases with the log, the arrhythmic mean quickly becomes useless to generalize a population of events, as a right hand skew causes even more exaggeration of the mean. So, to combat this, geometric mean MFI is often used to account for the normal or the log normal behavior of the flow data. I
Yeah,
don't know.
even median. I go for median fluorescent intensity because the median is less affected by outliers.
Yeah.
If you gate right, you won't have any outliers. You get to make up the gates.
Yay! That's the best part about flow. So it's so subjective.
Yeah.
It's great, but yeah.
Okay. So after well, confirming that those DC cells were probably more mature, they decided to check their functioning. So they co-cultured those DCs isolated from aged Elgal's knockout and wild type mice with cells, CD4 plus T cells and CD8 plus T cells isolated from young wild type mice. And first we can see they assessed the proliferation, of those CD8 plus cells and CD4 plus cells. And there is actually not much of a difference between wild type and L-Gauss1 mice.
Do they have a graph that quantifies this? Because I can never see any differences just based on the CF.
Well, they actually, for this experiment, they didn't include a graph. But, well, they said there was no difference. So I guess if they wanted a difference, they would just make it. It's true that there was no difference. So let's assume they are not lying. And after that, they checked cytokine production in the cultures. So we can see that for CD4 plus cells, the production of interferon gamma is elevated in the co-culture that used DCs from mutants, and the production of interleukin-10 is much lower. And for CD8 plus cells, there was no significant difference. Okay, and they also checked those co-cultures for T-Rex, and what they found was that in co-cultures with DCs from mutants, the percentage of T-Rex was significantly lower than in wild types. And they also decided to check for T-Rex and CTL4 plus cells in submandibular lymph nodes in L-Gals mutants and wild type mice. And also in those submandibular lymph nodes, the percentage of T-Rex and CTLA4 plus positive cells was lower. So these outcome, well, let's say in mice, maybe maybe because of those CD11C plus disease, I guess. So disease may alter the percentage of T-Rex and CTLA4 plus cells. Okay, so as they didn't find any difference in proliferation in those first co-cultures, they decided to use cells and media from this co-culture of CD11C and CD4 plus cells in the culture with CD8 plus cells from young wild type mice. And what they found was that proliferation and replication index in the cultures using mutant disease was elevated compared to wild type mice. So we didn't see it in the previous experiment, but when using more complicated microenvironment, this proliferation was altered. Okay, after that, they decided to check those NOD mice and bulb C mice as control for the expression of galectin-1 in serum and L-Gals1. And what they found was that the expression of both protein and mRNA decreased in NOD mice with age, and in bulb C mice, they didn't see this tendency. Okay, so they saw that those more prone mice to autoimmune diseases have reduced expression when they are old. So they thought that, okay, maybe and they also developed this syndrome. And so they decided to treat them with recombinant galectin-1 to check if it changes cell infiltrates in salivary glands. And they found that after the treatment, the percentage of infiltrating immune cells is lower. Among them, CD4 plus cells decreased, but not CD8 plus and CD19 plus. I mean, this is not significant, but when we see this CD19 plus, it's quite decreased, but it's not significant, so we cannot say.
Yeah,
for sure.
so they are using the error bars are standard error of mean, not standard deviation, and that's why it looks like they're not that big. But if you convert that to standard deviation, they're pretty big.
Yeah, that's probably true.
I usually present stuff as standard error of the mean.
I think that's I don't think it's accurate to present an SEM because it tells you something else about the what's the word, the variability of the data.
Okay, so generally we can see that treatment with galectin-1, well, let's say helps these NOD mice, at least just a little bit. Okay, and the last thing they did, and I think it's one of the most interesting for us humans, is they checked human biopsies. So they stained those tissue parts with anti-galectin-1 antibodies. And what they found was that in those Sjögren syndrome patients, this expression was reduced. As we can see here, this, well, let's say orange, I think, areas are much smaller than here in controls.
Just for a second, do you know in the previous experiment how did they administer the RGAL? Because in their methods they are citing another paper. That's how we administered it, and I'm in the next paper. I'm still not able to find out how they administered it.
I'm not sure, to be honest.
How can it be so difficult to just mention how you administered that in your own paper without citing?
As previously described.
Administer it intravenously, but it's sometimes different with mice. So I'm not sure.
Yeah, I'm hoping there's something very complicated that they didn't want to add to their materials and methods.
Yeah, sometimes you need to read like 20 articles to know how they did something.
Yeah, they just keep passing it on to the other article, one after the other.
And you end up reading article from like 1910.
Yeah.
The first method they use. So,
where they just did it and they didn't really describe it or whatever.
Yeah, because nobody cared at that time, and
yeah.
And actually, you end up reading article in German because back then English wasn't that common in science.
Yeah.
Okay, so generally what we see is that galectin-1 expression is lower in patients with Sjögren syndrome. And what they could also do was they wanted to check if this expression correlates with cells that infiltrate the tissues. So they stained those tissue samples for CD8, CD2, and CD4 expression, and they found that there was a negative correlation between CD8 plus expression and GAL1 plus area. And for others, this was not significant. So they concluded was that this expression influences CD8 plus cells infiltration.
Yeah, that's borderline significant, but still good for something in human studies.
Yeah. Yeah.
I like how
I
they
guess they
eventually came to the humans biopsies. That makes this paper complete. Like
It
all
took
of
it.
yeah.
It took it from some lower level journal to a PNAS.
yeah.
Yeah. And also I think that that's like um the biggest thing they could do with humans, because well they couldn't just take out their lymph nodes or things like that, because with humans it's you need to have a doctor to do some biopsies and things like that, and for most of the scientists it's not possible. It's not possible because we don't always cooperate with doctors.
Yeah, you need a collaborator. You need a
Yeah.
lab, like a
Yeah.
hospital which specifically has a lot of Sjögren syndrome patients who
Yeah.
do get biopsied. It's so many
From the
variables
labial
involved.
too, right?
What?
This is from the labial, it says. Biopsies from the,
Okay. It's from labial biopsies. Okay, so not
so
from
from the lip area, right? I
salivary.
wonder how big these biopsies were actually. And then when I'm learning from taking biopsies and kind of comparing pathology versus non-pathological portions or tissues, is that I think you get more. I think you would rather get more impact when you compare like non-diseased area to diseased area. You know what I mean?
Mm-hmm.
Like we've done biopsies on the skin before, but we've taken actual disease skin versus non-disease skin to kind of really show the heightened impact of what we see
Mm-hmm.
when we do stains like this. But the lip, oh my gosh, like
Mm-hmm. Yeah. Don't make me imagine that, please, Antia.
Okay, so this was the last thing with results. So maybe we can go to conclusions.
cool.
So I think one of the most interesting conclusions is that immune system functioning changes with age, which is also quite logical, but
Mm-hmm.
one of the conclusions. Because as we see, mice that as they were young didn't develop any disease. With age, it changes. And we can see that also in humans. As I said, like RA is also more common in people after, I don't know, 50 years of age than in young. Also, yeah.
While we are at this point, have you guys heard of these cells called ABC, age-associated B cells?
No.
So that's it. I think it was also recently found in a murine model of lupus, but these B cells, they are not present in young mice at all, and they show up in older mice after, I think, after 8 or 10 months. And these B cells, just these specific B cells, they are uh, they if you transfer them to a regular a healthy mouse, they get disease. So it's a very specific type. And yeah, that's we don't know how they're coming, how suddenly they emerge out of the blue. But yeah, these are, so if they are coming out with an advance in age, probably yeah, something's happening to our immune system, right?
Yeah.
Even the thymus thing, the thymus shrinking and having less T cells. But I would think that less T cells in that case would mean you have a little immunes, like you have fewer immune responses. But it turns out here you have more chances of developing autoimmunity and inflammation.
Yeah.
What's woozier?
Oh man, wowzer, stupid autocorrect. Oh, can I just add something here though? When we
Mm-hmm.
talked about immune systems functioning changes with age, yeah,
Yes.
yeah, we know that things like the thymus shrink in size as you get older. We know that immune cells age and kind of function a little bit differently. But with all these studies that people do, can we quantify maybe in increments at what age these age-related changes will occur in immune system? You get what I'm saying? So it's like you'd expect by 3-4 years of age for a child to, no, let me take that back, 8 or 9, 10 years of age, 6, 7, 8, 9, 10 years of age for a child to lose their first, grown teeth, right? So it's like then after a while you'd see wisdom teeth start to pop up. Is this something that we can quantify in immune cells and aging? Like, I just feel like sometimes it's kind of left up in the air to go against old versus young, and it's like to the extreme spectrum.
Mm-hmm.
Mm-hmm.
I think that one of the things is that when you're talking about age related, you're talking about a certain amount of stress over time. So it would have to vary person to person.
Right.
So we're talking about aged, but if you're under high stress and constant chronic infection and stuff like that, odds are good you're going to come to immune exhaustion if, as opposed to someone who's otherwise healthier maybe.
Yeah, but you would think with all the, at least like understanding that we have so far, then that leaves the same questions open for what is your control group for, disease state patients. It's like, okay, yeah, they don't have lupus or they don't have Sjögren syndrome, but at the same time, could they have diabetes or could they have, arthritis? Could they have something else? So it's like, it seems like we could do a bit of a better job at characterizing these incremental changes, at least for non AB variable people. Like, I don't know. I just feel like it's something a bit more we can.
You would
Yeah,
need
I
a
think that,
very large scale longitudinal study where people are constantly looked at for some parameters, maybe after 5 years, another 5 years, another 5 years,
Yeah,
and
why not?
yeah, that's a lot of money, right? Who
yeah,
that's a
Hey,
lot
but
of
why
funding.
not though? Think about
yeah,
it.
like
But
you
that's a
We
good idea.
mentioned,
That's a very good idea.
have the Human
yeah,
Genome Project. We have all these, even with the vaccinations and the medicines that are on the market today, takes 20
yeah.
years. You know what I mean? Like in some cases. So it's like, why can't we make a human immunological project and do something like this where this is more widespread? Like, I feel like there's just a lot of information we don't kind of know yet.
Yeah, it'll be multi-generational because we started right now, and if you even expect all people to live at least 80 years, and you want to continue it for 80 years, multiple generations of PIs will have to continue that project. So
I'm
it'll
fine with
be it'll be as it'll be actually bigger than the Human Genome Project because you will have to
that.
actually wait for people to get old right
Yeah.
from the birth.
I'm fine with that though.
Mm-hmm.
If you think about how we've started um, genetic disorders and passing it from generations, some of the first studies, even with like the Punnett square and who's the I can't think of his name, but
Mendel.
yeah. When they started these generational studies to kind of translate into human disease, and they saw that if you have a history of it in your family, then this is a likelihood, and so forth. So I just feel like we're smart enough to kind of have a better control that we can get more immediate answers than we do if you
Yeah.
really think about it.
Another
If you
another practical thing I can think of is if you have somebody, let's say you have 100 participants and you want to look at their immune phenotype for 80 years, people move out, they go to different places. Like
so
somebody can have a breakup, very bad breakup, and they don't want to be in your study anymore.
No, that's totally true. If you're interested in longitudinal studies, there's something called the Grant study, performed by Harvard Med, where they looked at happiness index of people over a 75-year period. So they started it in 1940, and they had been sending out questionnaires to people, I think every like 5 years or so. Let's see. Yeah, so there are tons of really good articles on it. where they're looking at like a happiness index. So this is interesting for a longitudinal study.
Yeah, when you have a survey or a questionnaire, that makes it easy, right, to have
Well, yeah, way
compliance.
easier than trying to get blood or biopsies
Yeah,
or anything else. But I
we want
still
your
think that,
lip. Give us your lip.
yeah, God. Yeah, so as long as you're not taking people's lips, I think they had over 400 people start in the cohort. So even if it's just a questionnaire or some other kind of thing, I think logistically it would be manageable if you had a large enough budget. And I think you're right that it's something that's super important. And
Yeah. I think long-term effects too. There was one study where they actually took maybe about 3 or 4 generations. Dang, I teach it all the time, like in intro biology. I cannot think of what the actual genetic marker they were looking at. But anywho, they took maybe about 3 or 4 generations of people in the same family. So there was a daughter, a mom, and a grandmom, and then maybe a great grandmom, something like that, where they kind of were able to map out just this prediction of what the future generation would potentially be in context of this genetic marker. And I just feel like, maybe there's something that we could get a better handle on, and 80 years isn't that long of a time if we think of just long-term humanity and how long we plan on staying on this planet. So it's like it's well worth it with immune disorders. Like, come on.
Mm-hmm.
Yeah. So when I was watching, I think I watched some little side documentary on that grant project, and they said that part of the hesitation at the first part was that the PIs that were starting it knew that they wouldn't get results, so they would have to deal with not having their results in their lifetime. And so that's just another hurdle when you're looking at these longitudinal studies that they, the postdocs that they were saying we're going to train postdocs that eventually would get data, after they're long dead.
It'll be a lot of money. And I also mentioned it could also have a little effect from politics, because when you have big studies, politicians want the results to come out within their term so they can use them to brag, like, hey, I funded something like this, nobody else does this. But if you have results coming 80 years later.
But also, there's still things you can get out of each increment, which is why I'm saying incremental understanding of these immunological changes within age. So it's still something you can get out of, in each component, of whatever time span each person is a part of. But I mean, the CDC has lasted how many years? The NIH has lasted how many years?
Yeah, they definitely have a study going. I would be interested in, over that longitudinal study, how our toolbox would change.
Yeah.
What we think is nice and fancy now with our flow cytometry, our 8 color panels. I mean, who knows in the next even 5-10 years what toolboxes are going to be.
How much advancement we could get just in those, like freezing down blood or freezing down plasma or freezing down, cells. We understand that very well. We can do that. So by the time technology changes in 20 years, we can go back and get more information out of it. It's just, to me, it's just something worth building on. So we need to all 4 put our heads together so we can make a business out of it ourselves.
Yeah, that could work. The 5-year increment thing, I think that's practical, and people are getting some results to show that they're part of it. It's not like they're not getting anything.
Yeah. Interesting.
Yeah, we could all be proponents of some kind of research like this. Like all 4. Okay. What else we can conclude from this study? What is also quite logical is that changes in glycosylation or glycosylation signaling may influence immunity. So this is what we saw, that if we don't have some receptors or ligands of these receptors, then some bad things can happen. But not only. I think that also in some physiological reactions of immune system, we can also see that glycosylation may influence them. Okay, and then that's galectin-1 signaling. So both the protein and its ligands are important for immune homeostasis. So if we disrupt it, then the immune system may go rogue. So let's hope it don't happen to us. And so also that in Sjögren syndrome, this reduced GAL1 expression may play a role, which is quite interesting as Sjögren syndrome etiology isn't fully elucidated. And we also can conclude that using recombinant galectin may act as a potential treatment for the syndrome. And it may be better than the treatments used now, so steroids or methotrexate or other similar drugs that well, they work, but they have many side effects. Oh, sorry. And what questions and what maybe further research is needed is that does galectin-1 directly influence PD-L1 expression? Because as you remember, this expression was lower in salivary glands of mutant mice than in wild type mice. So maybe it would be a subject to future research. Do you have any questions? No.
I'll start with the whole study plan. It was very well designed. I like the study, how they had these results. They had started from cells to mice using something they found as a treatment with the recombinant product, and eventually you also looked at human biopsies. Yeah.
And their focus on functionality, I thought was really good.
Yes.
Yeah. But
And
if
I
you
liked how they saw one thing in one result and they decided to check it more in other experiments. So they didn't only, keep to their initial plan, but also added some more things.
It was very logical, made a lot of sense, and this is way out of my wheelhouse, but I was able to follow it pretty easily. It was well written and well described.
Yeah, and well presented by Maria.
Yeah,
No,
excellent job. Excellent job.
Thanks.
Like, we still don't know so if these Sjögren syndrome patient have a deficiency in GAL1, we still don't know how it's coming to that. What's the reason for GAL1 deficiency? So there's still so much we don't understand about these genetic differences. Just I looked up, do you know phytohemagglutinin, that it's found in red kidney beans?
Yeah.
So if you don't cook them properly, you can have inflammation, because phytohemagglutinin binds to the sugars on T cell receptors, the other receptors, probably acts similar to galectin-1 in some ways. Oh no, not galectin-1. It actually does the opposite. I think galectin-1 is inhibitory. That thing stimulates. But after this paper, I will be reading more carbohydrates in immunity. I think I'm missing out on a big field.
Provide you with some more articles, as my PhD will be something about glycosylation and glycosylation patterns on probably on resting or activated immune cells, I guess, not in any diseases for now.
Yeah, and glycosylation has a direct connection with metabolism, because because the substrates for these glycosylations, they are glutamine and glucose.
Yep.
So if you have amino acid transporters in the cell, which pathways are working, all of that also affects glycosylation. It's such a huge web.
Everything is connected.
Yeah, so complicated. I wonder if something like this emerged through just random evolution. I have a feeling there is something more than that. How can something so complex just appear out of trial and error? That's my conspiracy theory, but I'll post that on a different page. Anyway, any more questions?
Nope. Excellent job, guys. Thanks so much for the discussion. And thanks everybody for tuning in.
Yeah,
Yes.
everybody. I'm
Anytime.
wondering, maybe we could discuss that later, but maybe we should also stream this on Facebook alongside YouTube, because a lot of people are there. But anyway, let's look at that. We also have to decide who will be presenting next in 2 weeks. I'll be ending this stream now. Thanks everybody for joining.